Today we can see about ribosomes and cytoskeleton. Ribosomes are granular structures which were first observed under the microscope as dense particles by George Palade in 1953. Okay, so ribosomes are small granular structures or dense particles which are present in the cell cytoplasm, isn't it? And what this ribosomes are formed of? It is formed of ribonucleic acid, that is RNA, and proteins. Okay, it is formed of ribonucleic acids and proteins. Several proteins join together to form the ribosome along with the RNA. Okay, now when we see this ribosomes, they are not surrounded by a membrane. So you can see the picture of the ribosome. There is no membrane surrounding the ribosome. So these are non-membranous cell organelles present in the cytoplasm. Okay, in eukaryotes the ribosomes are 80 years and in prokaryotes the ribosomes are 70 years. Both 80 years and 70 years ribosomes are having two subunits. One is larger and the other one is smaller. So in 80 years ribosome, the two subunits which are present are the larger one is 60S and the smaller one is 40S. If it is 70S ribosome present in prokaryotic cells, the subunits are 50S and 30S. So both the 80S as well as 70S ribosomes are formed of two subunits. Okay. Generally, the two subunits can freely exist, and at a certain times, that is at the time of protein synthesis, they will be coming together. Okay, and in this 80S and 70S, it is formed of subunits which is 50 and 30S or 60 and 40S. So here the S represents the Swedberg's unit. Okay, it stands for the sedimentation coefficient, that is the rate at which these ribosomes will be sedimenting. Okay, so S represents the Swedberg's unit which stands for sedimentation coefficient, and it is indirectly a measure of density and size. Okay, so this is about ribosomes. Next, we can see about cytoskeleton. Cyto means cell, skeleton means skeletal structure. So, inside the cell, there are skeletal structures. Okay, an elaborate network of filamentous proteinaceous structures are present in the cytoplasm and are collectively called as the cytoskeleton. So, cytoskeleton is formed of a network of filamentous protein structures. Okay, it is nothing but the skeleton means don't think some bony structure is there. Here it is formed of protein substances which are filament in nature. Okay, long protein filaments are there which forms the cytoskeleton. Okay, it performs various functions. It is involved in many functions like it can provide mechanical support, it can help in motility. Motility means movement of the cell, and it can also maintain the shape of the cell. So these are the functions of cytoskeleton. So what are they? It can provide mechanical support, motility, and maintenance of the shape of the cell. Okay. Next is cilia and flagella. The cilia and flagella can be considered as a part of the cytoskeleton as they are formed of microtubules. Microtubules means they are filamentous proteins. Okay, so we can consider these also as a part of cytoskeleton. Cilia is the plural form and cilium is singular. Same way flagella is plural and flagellum is a singular. So the cilia and flagella are hair-like outgrowths of the cell membranes. The cell membrane will be growing out as cilia or flagella in some of the unicellular organisms. Okay. And in multicellular organisms, certain cells also have cilia on its outer surface, like the columnar and cuboidal cells, which when they have the cilia on its outer surface, it can be said to be as the ciliated epithelium. Okay. Okay. Same way, the cilia and flagella are hair-like outgrowths of the cell membrane, which is seen in the prokaryotic cells and some of the eukaryotic cells also. Okay. Cilia are small structures when compared to flagella. Cilia are shorter, isn't it? They are small structures and they work like oars. Oar means water. It is the structure which is used to push the water backwards and move the boat forwards. In boat it can be used, isn't it? Same way here the cilia will be working like that of the oars. So when it works like that, it can help in the movement of the cell or the fluid. If the cell is moving, it can be moved forward, and when the fluid is moved, the fluid can be moved backward. So the cell will be moving forward. Okay. Now when we see the flagella, they are comparatively longer than that of the cilia. And this flagella is also responsible for cell movement. Okay. And in prokaryotic bacteria, they also possess this flagella. Okay, the prokaryotic bacteria also possesses this flagella, but this flagella is somewhat different from that of the flagella of the eukaryotic cell. Okay, the prokaryotic flagella is structurally different from that of the eukaryotic flagella. So now here in the picture you can see the flagella and the cilia. So this is the flagella which is longer, but here you can see the cilia which is shorter. So the direction of its movement, how it will be moving, as a result the cell can move. That is shown in this diagram. Okay. When we see the structure of this cilia or flagella under the electron microscope, we can see that they are surrounded by a plasma membrane. The cilia and flagella are covered or surrounded by a plasma membrane. Okay. The core of this cilia or flagella is called the axoneme. What is this core? Core means the central part of this cilia or flagella. Okay, that is the part which is present inner to the plasma membrane. Okay, so that is said to be as axoneme. Okay, so what is axoneme? The center part of the cilia or flagella is called the axoneme. Okay, and this axoneme possesses a number of microtubules which lies parallel to the long axis of the cilia or flagella. Okay, the axoneme possess a number of microtubules which run parallel to the long axis. What is this microtubules? These are very minute tubular proteins. Okay. Now, this axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules. You can see in the picture, there are 9 pairs of what is present? 9 pairs of radially arranged microtubules are present. Peripheral microtubules are there, isn't it? So here in this picture, you can see that what is present. These are the I think you are able to see that these are the what pairs of 9 pairs of doublets of radially arranged peripheral microtubules. These are peripheral microtubules. Okay, peripheral means what? Towards the outer side, outer region, it can be seen, not outside the plasma membrane, towards the outer side of the axoneme, the microtubules are present. Microtubule, I told you, these are proteins, isn't it? So the axoneme is having 9 pairs of doublets. Okay, see why we say it as doublet? You can see here each one is seen in 2 in number, isn't it? And when you count it, you can see 9 are there. This is 1, 2, 3, 4, 5, 6, 7, 8 and 9. So we can say that 9 pairs of doublets of radially arranged. These are arranged in radial position, isn't it? Radially arranged peripheral microtubules. Okay, and In addition to this 9 pairs of radially arranged microtubules, in the center we can see two centrally located microtubules. Okay, a pair of or two, we can say a pair of or two centrally located microtubules are also present. So this type of arrangement we can say it as 9 plus 2 arrangement. So what is this arrangement? It is 9 plus 2 arrangement. Why it is 9 plus 2? Outer side or in the peripheral side we can see 9 pairs of doublets are there, and in the center two centrally located microtubules are there. So such type of arrangement of axonemal microtubules is referred to as 9 plus 2 arrangement or 9 plus 2 array. Okay. Now, the central tubules are connected by bridges. You can see here the green line which passes in the center, that is the bridge. Okay, it connects the central tubules and is surrounded by, it is enclosed by a central sheath. This red line is the central sheath. Okay, it will be surrounding the central microtubules. Okay, and this central sheath is connected to one of the tubules of the peripheral doublet. You can see here the line which is joining one of the peripheral doublets, isn't it? In everywhere you can see one of the peripheral doublet is attached with the central sheath. Okay, it is attached by means of radial spoke. The line which you are seeing is called what? A radial spoke. Okay, so the central tubules are connected by bridges and is enclosed by a central sheath, and this central sheath is connected to one of the tubules of each peripheral doublets by the radial spoke. Okay, so how many radial spokes are there? There are 9 radial spokes. Okay, now the peripheral doublets are also interconnected by linkers. Okay, they are interconnected by structures called linkers. These are also proteins only. Okay, both the cilium and flagellum will be emerging out from a structure called the basal body. It is similar to that of the centriole. Okay, so both cilia and flagella will emerge out, will come out from structures called basal bodies, which are similar to the centrioles. Okay, next is the centrosome. Centrosomes are the organelles which are present in animal cells, and generally they are absent in plant cells. Okay, especially the centrosome is formed of two cylindrical structures called centrioles. Okay, so this centrioles, actually this centrioles was discovered by Van Benden, and its structure was given by Boveri. Okay, and these centrioles are absent in prokaryotic cells and even in fungi as well as the higher plant cells, and it is present in all the animal cells, especially all the mammalian cells contain centrioles except the mature RBCs. Okay, so the centrosome is an organelle which is made up of two cylindrical structures called centrioles. So you are able to see the picture here, a picture of the centriole. Okay, it is long cylindrical structure. This is the centriole, which is cylindrical in nature. Okay, and these centrioles are surrounded by an amorphous pericentriolar material. What do you mean by that pericentriolar material? Peri means outside. Okay, so outside the centriole there is a material called the pericentriolar material. It is nothing but it is formed of a dense mass of protein. What is present? It is formed of a dense mass of protein. So that protein will be forming an amorphous pericentriolar material. Okay, both the centrioles in a centrosome will be lying perpendicular to each other. Okay, which will be giving the organization like that of a cartwheel. Cartwheel, you may be knowing, you might have seen the bullock cart with the two cartwheel, isn't it? So the two centrioles in the centrosome will be lying perpendicular to each other, so that they will be organized like that of a cartwheel. Okay. Now when we see the structure of this centriole, we can see it. So here you can see the structure of this centriole. It is made up of 9 evenly spaced peripheral fibrils. So in the cilia and flagella we have seen peripheral 9 doublets are there, isn't it? But here it is 9 evenly spaced peripheral fibrils are there. How many are there? If you count again, here you can see 9 will be there. Okay. Here 1, 2, 3, 4, 5, 6, 7, 8 and 9. So we can say 9 peripheral fibrils are there, and these 9 peripheral fibrils are formed of the protein called tubulin protein. So which protein forms these peripheral fibrils? They are the tubulin protein. Tubulin protein means nothing but tubular proteins. Okay. So the 9 peripheral fibrils are formed of tubulin protein. Okay. And each of this peripheral fibril is a triplet. You can see it is a triplet. 3 are there, isn't it? Any one you see. Every one are a triplet in nature. But in the cilia and flagella we have seen it as doublet. Here it is triplet. Okay. And the adjacent triplets are also linked with each other. You can see they all are connected with each other. Okay. They are linked, and the central part of the centriole is also made up of protein. So here this is the central part of this centriole, and it is also made up of proteins, and it is called as a hub. What is the name given to the central part of the centriole? It is called the hub, and it is made up of proteins. Okay. And this hub is connected with the tubules of the peripheral triplets by radial spokes. You can see here it is connected by means of what? The radial spokes, just like the cilia and flagella. Here also it is connected by means of what? The radial spokes. So these are the radial spokes. You can see from each one of that triplet. Okay, from see, triplet is formed of 3, isn't it? In that any one is attached with the hub by means of this radial spokes. Okay. Now the centrioles form the basal body of cilia and flagella. Okay. Already I told you in the previous slide that the cilia or flagella will be arising from the basal body, which is similar to that of the centrosome, isn't it? So the centrioles form the basal body of cilia or flagella, and also it forms the spindle fibers that gives rise to the spindle apparatus, which is formed during the cell division in animal cells. In animal cells, when we see the cell division, during cell division, the chromosomes will be attached by means of the spindle fibers. Okay, the spindle fibers which is formed from the centrioles will attach with the chromosomes. Okay, so that it can pull that chromosome towards its end. For that it is used. Okay, that you will be studying in another chapter. Okay, so this is about the centriole. Okay, so in the when we see the structure of the centriole, it is made up of 9 evenly spaced peripheral fibrils, which is made up of tubulin protein, and each peripheral fibril is a triplet. Okay, and the adjacent triplets are connected with each other. So we can say it is linked. The central part of the proximal region of the centriole is also made up of protein, and it is called the hub. And this hub is connected with the tubules of the peripheral triplets by radial spokes. Okay, these radial spokes are also made up of protein. Okay, the centrioles form the basal body of cilia and flagella, and the spindle fibers that give rise to the spindle apparatus, which is formed during the cell division, especially in animal cells. Okay.